In this lesson, we get to take a look at Gregor Mendel's groundbreaking work in genetics. All right, so this lesson is our introduction to genetics, and there's no discussion of genetics that is complete without talking about kind of the forefather of modern genetics, Gregor Mendel. We're going to learn a little bit about him and his experiments and how they kind of laid the groundwork for everything we know about genetics today. Well, not everything, but most things. It did lay the groundwork for everything. We've learned a lot more since then. But anyway, let's get into this. All right, so back in the early 19th century, no one could really describe exactly why offspring of plants and animals looked like their parents. And nobody could really explain why traits seemed to appear and disappear and then all of a sudden reappear again down through later generations. So Gregor Mendel comes into the picture and helps us put some of these pieces together. Now, Mendel was a monk. He was born in 1822, and he spent a lot of his time in a monastery garden where he was able to carry out all of these experiments into inheritance and how that works. So he chose to work with peas, which are small and they're really easy to grow, and they produce a large number of offspring. So they were kind of like some of the best types of plants to use for what he wanted to study. So, why do you think inheritance studies that use peas is preferable to maybe using a species like pigs or horses? Well, the plants that Mendel started out with were what we call true breeding strains. Now, true breeding strains are created by basically taking two individuals that have the same traits and crossing them. And when you do this, all of their offspring are also going to have the same traits. Okay. And the continuation of these crosses constitutes a true breeding line or a true breeding strain. So Mendel and his different groups of true breeding plants would end up producing the same set of traits in each generation. And in order to figure out how the traits were inherited from one generation to the next, Mendel then took individuals from his true breeding strains and produced hybrids. So he had one true breeding strain here that had the same characteristics generation after generation. He took another true breeding strain that had different characteristics generation after generation, and he crossed them. He cross pollinated them so that they would create basically a new strain, or he would at least see what happens with the traits. Some of those traits included things like the color of the flower on the blossom. Some of them dealt with the color of the pea pods themselves, kind of the shape of the pea pod, if it was wrinkled or smooth, that kind of thing. So these are some of the traits that he was able to kind of study. And when he crossed uh, two pure breeding strains, he created what's called a hybrid. Okay. So a trait is basically any specific characteristic, and as I said, they could be anything from seed color to the height of the plant to the color of the blossoms and things like that. A hybrid now is, again, a cross from the true breeding individuals being merged together. Now, the general process of pollination in plants was pretty simple. Basically, he would take a little paintbrush and he would collect some of the pollen from one of the flowers on one strain, and he would then kind of brush that pollen onto the stamen or the pistil. I forget which one it is. It's either the stamen or the pistil um, on the other true breeding strain to make the cross. Now, why did he get the pollen and why did he brush it on the end of the stamen or the pistil? Well, because those are the male and female parts, as you can see clearly here. So inside is the female part where there are seeds found, and then these, actually these are the stamens. They actually produce pollen, which are basically the male cells, gametes, the sex cells. Okay, so kind of like sperm, but it's just pollen. It's not sperm. It's different than sperm, but the same idea, right? So you have the male pollen and the female ovaries. When the pollen moves down the, I believe this is the pistil, if I'm remembering my plant anatomy right, it would move down the pistil and enter the ovaries where it could then fertilize one of the eggs. Okay. Now, this is a process called, when he was brushing the pollen onto the pistil, that's a process called pollination. Fertilization is different. Fertilization is once the pollen reaches the egg, or in this case, the seed, it can then become a fertilized cell. Okay. So fertilization and pollination aren't exactly the same thing. Now, some might begin to think that self-pollination seems like a form of asexual reproduction. And while you have offspring that are produced from only one parent, each offspring forms from the fusion of the male and the female cells. So with a true breeding strain, you would be pollinating strictly with the pollen from the same plant. Okay. So you would end up with the same characteristics. You wouldn't have any difference in the DNA because it's all the same plant. But if you were to cross two different strains, the way that he did, you're going to end up with some changes in the DNA. There's going to be a new combination of DNA. So don't want to put the cart too far behind the horse. You don't want to put the horse in front of the cart, or the cart in front of the horse. I don't know. You don't want to do that. All right, let's move on. All right, so as you look at these two flowers, we've got the pollen being cross-pollinated from one strain to the other. What do you notice about the flowers that are shown here compared to the flower on the last slide? Let me go back to the last slide. Okay, do you notice anything different? Okay, well, if you say, well, it doesn't look like this flower, let me get my little laser pointer, it doesn't look like this flower has a pistil and the ovaries. It looks like it only has the stamens that produce the pollen, and this flower doesn't have any stamens to produce pollen. It only has the pistil and the ovary. So yeah, it's different. If we look at the last slide, it has both. Okay, so one flower, the flower in the previous slide, has both male and female structures, but here the flowers only have either the male or the female structures, not both. So how did this happen? Well, actually, Mendel actually removed reproductive structures from the pea flowers so that only one set remained. Now, why do you think Mendel was trying to what do you think he was trying to prevent by removing one set of the reproductive structures? And why? Well, it comes down to the fact that he was trying to prevent the flowers from self-pollinating. He wanted to be able to control, because again, he's doing experiments. You have to control things, right? So he wanted to make sure that he could control which plants were being crossed so that he would be able to keep track of what's what and who's who. Okay. All right, so here are some pea characteristics, and Mendel looked at 7 different characteristics that you find in pea plants. And his goal was to look for patterns in those traits across generations. Now, in Mendel's day, of course, the terminology we use of genes, alleles, and chromosomes, that was all unknown. They didn't use those terms. But he knew that some plants produced a round seed and some produce a wrinkled seed. Some of them were yellow, some of them were green. Some of the plants had purple flowers, some had white, some had a smooth pod shape, some had constricted or wrinkled. The pod color could be either green or yellow, depending on the strain. And the flower position, where the flower was located. Was it located in the middle of the plant or the bottom of the plant, or was it located up at the end, very tippy top of the plant? And also plant height seemed to be consistent across generations as well. So those are the 7 different characteristics that he looked for in his plants, and he used combinations of those to cross the different strains to see what would happen. So some important vocabulary words that we need to commit to memory so that we know what's going on when we're talking about it is genes and alleles. So genes are passed from one generation to the next, and they basically determine what an individual looks like and so forth. Whereas alleles are just the different forms of a gene. So the gene could be the seed shape, but the alleles could be a round seed shape or a wrinkled seed shape. Okay. So the gene itself is just the characteristic that we're looking at. The allele is the differences in that particular gene. Okay. So Mendel's first conclusion from his experiments was that an individual's characteristics are determined by factors that are passed from one parental generation to the next. This was new thinking. And so that was kind of a conclusion that he ended up coming to in his experiments. Now, today we can talk about genes that govern particular characteristics and the different alleles that control the different versions of those characteristics. But for Mendel, he could consider only really the traits themselves. So what he called factors, today we call those genes. And the different versions of a gene that produce the different traits, the alleles, for example, yellow seeds versus green seeds, we call them alleles. He didn't have a term for them. All right. So here we have some questions, and we need to fill in the blanks. And so these are some constants that we have to keep in mind as we study the things that Mendel was studying. So characteristics, and this is just going to kind of help with our understanding of genetics overall. So these are some basic premises that we operate by when we talk about genetics. So characteristics such as seed color are determined by what we call genes. He called them factors. The yellow and green seed colors are determined by, what do you think? Think about it. If you said alleles, you would be correct. Now, there's something we haven't talked about yet, and that is that some alleles are dominant and some are recessive. So we'll get into what that means as we move along, but just know that in this particular case of seed shape, the allele for yellow seeds, and so actually seed color, not seed shape, the allele for seed color, yellow, is the dominant allele to the green allele. So if you were to cross a dominant yellow and a recessive green, the offspring would be yellow. Okay, we're gonna dive into that and kind of pick that apart as we move into deeper into the genetics study. But that's kind of a good little start on it. So let's talk about the principle of dominance. But before we do that, imagine that you cross 2 plants, one with green seeds and one with yellow, and the offspring of the plant produced only yellow seeds. What could you infer about the green and yellow seed color traits? Well, we already kind of answered that. What we could infer is that the offspring are going to express yellow seeds. Now, how would the results of the cross change if green seed color were dominant to yellow seed color? Well, in that case, the offspring of a green-yellow cross would have green seeds. Whichever one is the dominant is going to dominate and be expressed and be seen. Now, sometimes students get the impression that when you're considering traits like pod color or seed color or even seed shape, that the structures themselves are somehow being crossed. So really, I need to remind you that these structures are traits of full plants and that the results of the crosses refer to the traits shown by the offspring plant, if that makes sense. All right. Now, what is this principle of dominance? Let's talk about it for a minute. So this is actually Mendel's second conclusion, the principle of dominance. And basically, it tells us that some alleles are dominant and some are recessive. Okay. And as I just explained, dominant means it's going to be displayed in the offspring over the recessive version of the allele. Another principle of dominance says that an organism with at least one dominant allele will exhibit that trait. So you're going to find out that there's actually two alleles for each gene. Mendel also determined that an organism with a recessive allele will only exhibit that trait in the absence of a dominant allele. So again, that dominant allele is going to dominate if it is present at all. Okay, that's important to know. All right, so in this table, we have Mendel's crosses and everything that he did, and we have what's called the F1 generation in the bottom row here. See that here? But these are the crosses for seed shape that he did. So this is the parent generation. He crossed a round and a wrinkled, and he got a round. He tried seed color, yellow and green. He got yellow. He tried flower color, purple and white. He got purple. He took pod shape, and so forth and so on. You get the idea. So what did that tell Mendel about traits and about dominant and recessive? Well, it told him that round seed shape was dominant to wrinkled seed shape. It told him that the yellow colored seed was dominant to green colored seeds, and purple flowers were dominant for over the white flowers, and so forth and so on. So basically, from the parental generation to the first offspring, the F1 generation, he was able to determine which alleles were the dominant alleles. Okay, now you might also notice that these offspring down here in the F1 generation only showed the characteristic of one of the parents. It didn't show both. Now, we're going to find out once we learn more about crosses that there are traits that have a mixture of both, and there's all kind of rules and things like that that'll help us kind of keep them straight. But for now, really what's important to understand is that there's a dominant allele and a recessive allele. And each gene has two alleles. Okay, so if the offspring gets two dominant alleles, it's going to show the dominant trait. If it gets two recessive alleles, it's going to show the recessive trait. And if it gets a dominant allele and a recessive allele, one from each parent, it's going to show the dominant trait because the dominant exists. The dominant is present in the offspring. Okay? Now that's it for this lesson. In the next lesson, we'll get into segregation. So we'll see you then.